So
You
I'm
deserve
gonna,
it.
yeah, it's always embarrassing. So I thought I'd tell you, this is what the seminar will cover. And so if you space out and start reading your email, which I have to admit, sometimes I do, like pay attention right now and you'll get the gist of it, and I'll try and wake you up for the high points. So I'm gonna go over the logic for repurposing. I think it's maybe too obvious to go over, but I think it's worthwhile doing. So why are we focused? Why are we and everybody else focused on repurposing? And I'll also try and allude to some of the problems of repurposing as a strategy. I'll take you through this mechanism-based approach that really Nevin has been leading, and we've joined. He was very early, and this is, it's a career built upon this, but in particular with COVID-19, they started this work in mid-January, so they were ahead of the curve, and we only started getting involved in a month later. If we have time, I've tried to present this work to just internally twice before, and I never get beyond the first two panels. But if we have time, I'll go over some of the chemoinformatics that supported it, and how, what we see as the future for that. And I'll tell you one story that isn't exactly to do with it about COVID-19, but it illustrates the use of the methods, and it's coming out in Science, so I wanted to talk about it. And then we're also using a docking-based approach, not just repurposing, but also de novo ligand discovery using the ultra-large libraries that we've become very excited about, these virtual libraries, if I have time. So that's the overall trajectory of the talk. So I wanted to just to go over, everybody is familiar with the drug discovery pipeline, and we've all seen it so often that it blurs, I think, and it's really just, it's mostly used as a metaphor, right? But I thought I'd give you an example of one case of actual drug discovery that led to an actual drug in the space of antiviral for Hep C, for hepatitis C virus, HCV. And this is the drug Daclatasvir. And that was introduced a few years ago by Bristol Myers Squibb. And it, like every drug discovery story that I'm aware of, has a huge amount of innovation. It's tried to fit into a particular schema when people are telling about it, but in fact, they're all different, and they all rely on several miracles happening. And the Daclatasvir was no exception. It started, I think, in early 2000s, and they did a screen, which sounds very normal. They don't exactly say how many, but over 1 million molecules were screened. But they did it in this very innovative way. The screen had a lot going on with it. They were looking for molecules that inhibited the RNA part of viral replication of HCV, but they weren't doing a direct target binding assay. It was a phenotypic screen looking at infectivity, and the way they looked for infectivity was, I don't have time to go through it, it was very innovative, and they had a lot of controls built in to get to a new target. And anyways, after a lot of work, they got one confirmed hit, which is this molecule shown here, which was quite potent in the antiviral assays. And because of the counter screening that was built into this amazing assay, they were confident that it was working on the virus. But for those of you who have a medicinal chemistry background, you can look at this central core. I don't know if you see my mouse, but if you look at this central core ring, it looks like it might have redox issues, electron cycling issues. And I don't know if they were aware of that or not, but sure enough, problems emerged. It turns out that this compound was not the active compound. The active compound was a dimer, that formed in DMSO over time. And it took them, I don't know how long to figure that out, but normally it would have sunk the project, right? They got one hit, and the one hit they got wasn't what they thought it was. It was a dimer, of a covalent dimer of the what they thought was the material. But they deconstructed it and reconstructed it and got to this new molecule that represented the dimer and that was active and that they could do structure activity on, and were able to progress it to better and better activity and less and fewer tox liabilities to this molecule, which is the actual drug, was the candidate. And then they took it into, 5 years later, basically, they took that molecule into clinical trials with, investigational new drug application, and then phase 1, phase 2, phase 3 trials. And then in 2015, the drug was approved, and it's now used. I think it's doing very well for patients and for the inventor for BMS, and it's used in combination with another drug. But the point of this is that to get a new chemical matter acting, especially through a new mechanism, it's a long road, and this just illustrates some of the highs and lows of that road. So for COVID-19, for SARS-2, we don't have the luxury of a 12-year time horizon. And so much of the efforts that have been going on in the community has turned to repurposing. And there's, the literature is full of such papers. Some of you may be involved in those efforts. Some of you might be reviewing the papers, overwhelmed with reviewing the papers. There's some amazing ones, and some are of variable quality, I found personally. This is, I think, one of the really great ones. It's from Sumit Chanda's lab and others. It's in bioarchives. I think it'll be coming out actually in the formal journals soon, if it isn't already. And they did something really neat. They took this reframe library, which is actually, to me, a new library, but others might be more familiar with it. It's about 12,000 compounds. It's composed of approved drugs, investigational new drugs, and a few preclinical compounds. The actual numbers seem a little bit funny to me, because there's not 5,000 FDA approved drugs, unless they're using different formulations of the same drugs. There's more like 1,800. So the numbers don't quite add up. But anyways, it's a really interesting collection. And they screened this collection directly against these Vero cells, which are these African green monkey cells, which are widely used in the field. We use them too, as you'll see, for viral infections. It's a cell line. And they found molecules that inhibited viral infectivity without inhibiting cell proliferation, or no, that weren't cell toxic. And here are some of them. They have varying activity. Some look really good. I just wanted to show you these guys are a subset of the molecules that they describe. They're all the reason why they're drugs is that they have activity on drugs or candidates on GPCRs. Actually, I don't think any one of these 5 is an approved drug. They're all either clinical, have been in the clinic. Astemazole was approved, but it was withdrawn for hERG effects. It was the molecule that put the cardiac channel hERG on the map for cardiac toxicity. Unfortunately, it was developed as a second generation antihistamine. But anyways, it was an approved drug. The other ones are candidates, active, but with pharmacology active in people. But they all act on GPCRs. And what's quite, I think, pretty clear is that these molecules, as effective as they are on the virus, are not acting through their known mechanism of action targets. acting through another target, and what targets they might be working on is unclear. And, you know, at this time, and, you know, actually at any time, that doesn't stop a drug from going forward, not knowing its target. But it is a big barrier to understanding what it's doing, de-risking it, and optimizing it. So this is one of the challenges for repurposing, is not knowing mechanism. And it's not just a theoretical challenge, it's a pragmatic challenge. And so what I'm going to talk about, you know, for the rest of the talk is a mechanism-based approach to repurposing, and where that's taken us. And that's, you've heard from Nevin earlier. This is Nevin in the spectacular shirt here. I'm competing, people think I'm competing with Nevin, but my view is I got there first and he's competing with me. Anyway, so Nevin really organized this group. It's a big group at UCSF. These are all faculty that I'm showing. And I mean, I think one of the blessings of COVID-19 is that in former times it would be impossible to get all these people together in one place at one time. And we even failed this time, but so people stopped traveling, so we were able to take a picture together. And then social distancing happened, and so that now we can't even take pictures together. But anyways, it's a super talented group that Nevin really organized, and there's a lot of technologies involved. I'll highlight a few of them, but I think it's the center point of articulation is through the proteomics that Nevin group has been leading, and that's what I'll talk about next. So in the middle of January of this year, Dave Gordon in Nevin's lab got the CoV-2, the SARS-2 genome synthesized and expressed in HEK cells, HEK293 cells. And what they did is they individually heterologously expressed the 27 what they call bait proteins, which are the viral proteins in HEK cells, and they had engineered them with affinity tags on them. So each protein was affinity tagged, and they expressed them in the HEK cells, and then lysed the cells and pulled down the strep tagged viral proteins and any human proteins with which they were associated. And then they flew them in the mass spec and looked to see what human proteins were associated with each of the 27 individual viral proteins. And from that, they constructed this proteomics map. So what are shown in the in red at the center in these diamonds, these red diamonds, are the viral proteins, the 27 viral proteins, and then connected to them are the human proteins that they pull down. And that the ones that are colored in orange turn out to be drug targets, which I'll turn to in a second. And so they pull down with the 27 human protein. One was a mutant, so effectively 26 human proteins. They pull down, 26 viral proteins. They pull down 332 human proteins. I said 69, I think it's really 63 of them are druggable. And by druggable, it's a trivial definition of druggable. It's druggable for this purpose of the first paper, at least, were human proteins. So we're really focusing on human proteins for which there are known drugs or investigational new drugs, so molecules that are in the clinic, or preclinical molecules, advanced preclinical molecules. So that's our trivial definition of druggable for this project. And so what we did, and this is the work of Matt O'Meara, who until very recently was a postdoc in my lab and now has his own lab at the University of Michigan, Tia Tumino, Jiang Zhang, J.K. Liu, and Ximing Peng. What they did is they screened the established drugs, so approved drugs, the the investigational new drugs and what we call probe molecules here, so advanced clinical molecules. And the criteria for being screened was they had to be one in those classes, and they had to be commercially available. So there's more than 1,600 FDA drugs, but not that many more actually. But these are the ones that we could actually purchase. And there are more molecules have been into clinical trials than 20,000, but these are the ones you can purchase. And, you know, for this project, we needed to get the molecules. And so what emerged from this were 29 drugs, 12 investigational new drugs, so in the clinic, and 28 preclinical molecules for 63 targets. And the targets are shown here. I don't expect you to get that much out of this figure, other than they span a large range of biological processes. And one of the great things about the Gordon paper, you know, which I don't take any credit for, it's just great to have read it, is that they cover a wide range of functions, but when you drill down mechanistically, you can understand in many cases what the viral protein is doing in subverting the human protein to its ends. You know, the virus, I don't have to say this to this group, but it's, you know, it's useful to remember, you know, with a genome of 29 genes, 29, you know, gene products, obviously it's not enough to sustain life. So it has to subvert human proteins to propagate itself, and it has to subvert cellular mechanisms to prevent the cell from, you know, attacking it and giving the virus a clean, safe, well-lighted place to replicate and expand. And though, you know, and it's well understood that that's the major mission of viral proteins, to subvert the human proteins to allow it to replicate, and to subvert them or often, you know, ensure they don't express, to keep to reduce the antiviral response. Okay, so here's the 69 drugs that we prioritized for testing, which I'm about to get into, because, you know, it's a wide range of targets and a wide range of drugs that emerged. And this doesn't mean that much. It's just to show you the spectrum of molecules and how many were approved in clinical trials and preclinical. We then, none of this would have had the impact it did without the collaboration of Adolfo Garcia Sastre and his lab at Mount Sinai in New York City, and Marco Vignuzzi at the Institut Pasteur in Paris. And so we sent them the drugs, and what they ran this viral infectivity assay on them. Also, so the Viro6 cells that you saw in the Sumit Chanda papers are also, they also use them. They put the, you know, they grow up the cells for 24 hours, put the drugs on them, and then infect with the virus. And then that goes on. They allow the virus to expand at a relatively low multiple of infectivity, and for 2 days, and then they fix the cells and analyze the results. And in Paris, they looked for, you know, production of viral RNA using this quantitative RT-PCR reaction, and they also did these plaque assays. So how many viral, you know, active viral particles were produced. And in New York, they focused on this nuclear stain for this N protein, the viral N protein, and its location in the nucleus, which is correlated with viral propagation. And they looked at, you know, the compounds we gave them. They overall 47 of the drugs that we prioritized were tested at the time of this paper, and each one was looked at in full dose response, and they were, you know, checked for effects on the cells, the Viro cells themselves, the green monkey cells. And so it's an enormous amount of work that they did in the two labs. Okay, so two classes of drugs emerged. The first one, the more potent one, really, were these protein biogenesis inhibitors, including zotatofin, which is this elongation initiation factor 4A inhibitor. Also ternatin 4, which hits a later process in protein biosynthesis, and several others that hit other points in protein biosynthesis. This is, I'm showing here up at the, sort of at 12 o'clock on this slide in the center top, the dose response curves for zotatofin. The black curve is the cell viability curve. And when I initially saw this, I thought, oh, this isn't so good. It's killing the cells. But what, Kayvon, this is a compound that emerged from Kayvon Shokat's lab at UCSF. And what he points out is that the zotatofin isn't killing the cells. It's cytostatic. It's preventing cell proliferation, and that's sort of well known for these molecules. And the cell lines like the Viro E6 cell line is particularly sensitive to this. They often even don't have that effect on primary cells. The red curve is the dose response curve for infectivity, and it's the red curve is again percent infectivity on the left, and I think it's the green curve is looking at viral titer. And so the green curve is on a log axis, and so this little drop here right at the top is, I think like an IC90 actually. So it's quite potent. It's the IC90 is 37 nanomolar for this molecule, zotatofin. Okay, and those molecules are still being actively pursued, but I'm not going to talk about them. I'm going to focus on the second class of molecule that appeared, which are these sigma receptor binders. And the reason I'm focusing on them there's two reasons. The first is they're actually the class that we focused on, and the molecules that came out of our analysis. And they're I think the more novel target, a little bit more novel. There's precedence for these elongation initiation factor inhibitors, these EIF4A and others, 4E, to have antiviral effects. But the sigma receptors were really new. And so they came down at this NSP6 protein in the proteomics assay was associated with the sigma-1 receptor and others, actually. And NSP6 is known from work on other coronaviruses, SARS-1 and others, to be involved in the production of these dimembrane vesicles in which the virus is thought to propagate sort of in isolation from the rest of the cell. So it's known if you knock out this NSP6, that's deadly for the virus. And as you'll see, the role of sigma-1 in this is at least can be mechanistically understood. And sigma-2 came down with this ORF9C protein. The importance of ORF9C is more controversial for the virus. And actually the pharmacology, or at least the mechanism of sigma-2 is less understood. Sigma-1 is actually also, both sigma receptors are sort of dark horses in pharmacology. They've been around for, 4 or 5 decades in pharmacology, and both have been targeted extensively in drug discovery, but their actual roles in physiology and disease remain controversial, actually. Anyways, the sigma-2 receptor is thought to have a role also actually in membrane biogenesis and cholesterol homeostasis, among other things. So both have roles in membrane biology and protein stability. The sigma, more is known about the sigma-1 receptor. It has these chaperone properties and is activated under cell stress, and it traffics to it's localized in the ER initially, but it on stimulation it traffics to other organelles and is involved in stabilizing or certainly interacting with a lot of different proteins. And it's thought to be activated on cell stress, which is, at least sensible as a response to viral infection. And its structure is known. So the structure was solved in Andy Cruz's lab several years ago, and so we have a good like physical, structural biology understanding of the protein. Okay, so when we saw sigma-1, we've have a strong focus in GPCRs in my lab, and so especially for aminergic GPCRs, there's a strong overlap with the sigma receptor pharmacology. So we were very aware of both targets. And so we prioritized molecules that we thought would be potent against either or both receptors. And these are the molecules that first emerged and that were discussed in the paper. PB28 is a tool compound, preclinical compound, but very potent against both receptors, actually. And so what I'm showing you here are, just to take you through PB28, is the dose response curve for cell infectivity in red, and for the health of the vero cells in black. And so you can see that PB28 has negligible effects on the cells directly. It's not cell toxic or even cytostatic, actually unlike the protein biogenesis inhibitors, and it's pretty potent on the cells. So these are other drugs. Haloperidol, well-known antipsychotic. Clopirastine is a antihistamine and antitussive. Clemistine, same thing, antihistamine. So some of these are ancient drugs. And hydroxychloroquine, of course, is much in the news. And you can see that it's also active antivirally here. And you can see that, so this is another thing, that the fundamental mechanisms of the diseases that these drugs are used in, antipsychosis, antihistamine, anti-cough, vary quite a lot, as do their structures, but they all are active on the sigma receptor, and I'll come back to that in a second. And then these are these icing. So these were done. The curves I'm showing you are the nuclear staining assay that's run by Chris White and Adolfo's lab. But then sort of the gold standard are these TCID, these viral titer assays that they run. And in those assays, this is PB28 here at the top. And and I showed you that with zotatofin too. It's IC90, so it's once again, it's a log scale for plaque forming units, and so the IC90 of this compound is decent. It's like 280 nanomolar. So I'll just take you through a few other examples of the molecules that were published or that are new that we haven't published, so have come out since the paper came out. Dextromethorphan was in the paper. It's interesting because it's thought to be an agonist of the sigma-1 receptor. So this is this molecule is ubiquitous, certainly in the United States and Canada. In cough medications, it's an antitussive, anti-cough medication. It's thought to be a agonist of the sigma receptor. So in the US, it's really difficult to find cough medication that doesn't have dextromethorphan in it. If it does it, then it's codeine, right? Which has its own liabilities. And what we saw, what they saw in Paris, is that as you raise the concentration in this red curve of dextromethorphan, the it's proviral basically, viral infectivity went up. And this is on a log scale. So these are substantial effects actually. And what we tentatively suggest, and it's still tentative, is that this reflects the agonism. So this molecule turns on the sigma-1 receptor. Probably turning on the sigma receptor probably means having it go from its inactive oligomeric state to an active monomeric state or less oligomeric state. And so this is an example. This sort of gives it's a good counterpoint to the role of the sigma receptor. this is, I want to show progesterone. It's a very different class of molecule. It's a steroid. It also hits the sigma receptor. And we wanted to, it's an example of it's not even charged. It's not a cation at all. And yet it's not that active. It's, between 5 and 16 micromolar, depending on the day you do the assay. But it was, I think, a good illustration, speaking to mechanism. And then we have a whole bunch of other molecules that have emerged, and I'm not gonna spend too much time on them, just to say that, since the paper came out, we've been looking for more and more molecules, and we found them, amiodarone, lacrodar, and several others stand out because they're relatively potent. They're sub-micromolar, and they're, as drugs, they have pros and cons. Lacrodar is not an approved drug, and amiodarone, although it's widely used, has some. It's not a perfect drug, but we're interested in drugs at this point that have high potency, relatively high potency, and can get up to high concentrations in humans, and these drugs have that. It's, I think, important that, from a pharmacokinetic standpoint, to treat the virus, that they get localized to the lung, which we don't know about yet, and reach high concentrations. So, as you know, just like an antibiotic, you want to really slam the virus. So yeah, just to say, so a broad range of classes have been found, because we know the structure, certainly of the sigma-1 receptor. We can model with, I think, pretty good confidence how these guys bind, and if we want to start optimizing that, that's open to us. And so just going through some of the pharmacology here. So amiodarone here, for instance, gets to 30 micromolar on human dosing in the bloods. So I think that will be important. Okay, and so to finish up this part of the talk, this is sort of where we are. The molecules we've discovered to date hit a wide range of targets, at least as their mechanism of action targets. Lacrodar was developed, I think, by GSK actually as a PGP inhibitor, a P-glycoprotein inhibitor for cancer, but it also hits the sigma receptor. And amiodarone is a calcium and sodium ion channel antagonist, but it also hits the sigma receptor. And other of, the antihistamines hit the muscarinic receptor and the histamine receptor and the sig. So it turns out when you put all these together, that the consensus targets that emerge are the sigma-1 and sigma-2 receptors, which doesn't rule out other targets. And in fact, something that we're very energized about, like, are we really sure it's the sigma receptor? One of the things that worries us right now is that there's a discrepancy between sigma receptor potency, like when you measure on target, which we've done extensively in collaboration with Andy Cruz's lab and with Brian Roth's lab. That the molecules can be quite potent on the sigma receptor, or like low nanomolar or sub-nanomolar, or they can be just sub-micromolar, and there's not much difference sometimes in their the antiviral potency does not track linearly with their on-target potency. That could be for all sorts of perfectly reasonable reasons, like these are intracellular targets, so it could reflect the ability of molecules to get into the cell, but still it's a concern. What I can say, on the other hand, is that cationic, I don't show these data, but I should. We have other molecules that are in this same class, like antipsychotic class, but they don't hit the sigma receptor, for instance, like clozapine, and they're also inactive on the virus in our hands. And preliminary results suggest that RNA knockdown of the sigma-1 receptor at least reduces viral infectivity. but that also comes with caveats. So we're not fully confident of that result yet. So we're still trying to nail this down mechanistically. There's concerns about, well, maybe it's just endosomal acidification, like chloroquine, for instance, is a reagent that people use, forget about being a drug, it's a reagent that people use to block endosomal acidification and lysosomal, in that sort of endosomal internalization pathway, and which the virus uses. And it's plausible that these molecules are just basically buffering the endosome. We think that the time of addition studies that Adolfo's lab has done, so it turns out these molecules are equally antiviral, irrespective of when you add them to the cell. So if you add them later and later, they're still antiviral. And that suggests that you're not blocking the early stages of infection, which the endosomal pathway would fall into. And progesterone.
Very much.
Okay. So there's other types of competition assays, like just classic pharmacology that we can do. I mean, just classic pharmacology is no joke. And developing chemical probes to nail down the mechanism. And then the last point is new molecules for new targets. And that's, so, I told you that the sigma receptors and these protein biogenesis enzymes have emerged from the sort of the first shot on goals from these studies, and we're super excited about them. But there's other targets that are emerging, and we'd like to prioritize molecules for them. And of course, taking these molecules further into pharmacology in animals and maybe people. But guys, I've been at this for almost 45 minutes, and I don't know how much time I have actually, and I have a lot more material. But is this a good place to stop, or should I go on?
There's no firm timeline for you to stop if you'd like to keep going. I think we can go for another 15 minutes before we should stop and make sure there are no questions.
Yeah, I do want to leave time for questions. Okay, so maybe I'll just go through this one other story, which is around new molecules for new targets. So how do we get them? So up until now, we've been focused on the computational side of this story has been, embarrassingly trivial. We've just looked for knowns. That, required a certain amount of infrastructure, finding the knowns and understanding what they did and trying to be thoughtful about it. But it's nothing very sophisticated, algorithmically. But we think we've largely exhausted that route. So the next part of the campaign is being more systematic and comprehensive about it. So of the targets that emerged from the proteomics, we've focused on 6 or 7 where we tested more, but it was just a thin slice through things. And if the first drug didn't work, or the first 1 or 2 drugs, we dropped that target. And now we want to return to them. And so going forward, at least from my lab side of things, we're going to be screening the about 24,000 drugs and investigational new drugs and preclinical molecules that we've prioritized against the 69 targets. And we're doing it chemoinformatically using this similarity ensemble approach that Mike Kaiser developed when he was in our lab. He's got his own lab. And it's a ligand-based approach, and you just take each molecule and compare it against the ligand sets for the I say 69, it should be 63 for the ligands that are known to bind to each target, and say, well, how similar are each of the molecules of the 24,000 drugs to the known drugs for the human targets? Yeah, we're really focusing on the human targets. And then we control for random similarity. We use what we found, just like BLAST, we found that if you didn't control for random similarity, you'd be strongly biased towards targets that had more molecules, just like you're biased in BLAST towards sequences, longer sequences. So we used basically the BLAST statistical machinery to control for random. And so we get these z-scores and e-values, and that allows us to control for basically off-target similarity, just random similarity. And the method, has flaws. It's a ligand-based method. It's not a structure-based method, but it's been actually remarkably successful at predicting drugs for targets. And we have a series of papers about it. And this is where I take this is the detour away from COVID-19, because I wanted to illustrate pragmatically how it works with, a more recent story. So this is unpublished work, and it's actually it was started before COVID-19, I gotta say, well before, and it was on inactive ingredients. So we've done a lot of the papers cited below are on drugs, actual drugs. And it turns out that, as you for sure know, but maybe forgot a little bit, there's two components to drugs. One is what's called the active pharmaceutical ingredient, the API, and that's what you think of as the drug. What drug are you taking? Oh, I'm taking famotidine for heartburn. But within the drugs that you actually take, the formulations are what are called the inactive ingredients. And you can see this is just two different drug labels. And here's the inactive ingredients for famotidine, for instance, which is Pepsid. You get this over the counter. You don't even need a prescription. And here's the inactive ingredients that are listed here. And when we got involved in this, we were introduced to this issue by the FDA, who funded a lot of this work, and they were wondering, how to classify inactive ingredients. And so we started looking. We had never paid any attention to them, and we started looking at the molecules that were classified as inactive ingredients, and showing you two of them there. These are dyes, and
well,
our first thought was like, yeah, you know what? Some of these guys don't look so inactive. How do they know they're inactive? And so actually, it turns out that inactive ingredients, excipients is another name for them. The inactive ingredients in drugs are inactive by consensus as much as anything. Most of them, or maybe all of them, have been studied in whole animal toxicology, but they haven't been studied at a molecular level. They've been, a lot of these there's very few new inactive ingredients, because the FDA said, okay, this is our set of historical inactive ingredients, and it's fixed. And if you want a new inactive ingredient, that's no problem, but you actually have to do proper trials on them. These guys are most of the inactive ingredients date from decades ago, and have not changed. And they've been studied for gross animal toxicity, but, well, with some exceptions, not too many other things. So they're safe because they don't make rats sick over, a 30-day course, the end. And they're inactive. So we thought, well, let's just take that as a hypothesis, and let's take all of the excipients that are approved by the FDA, and some in Europe, and let's put them through this similarity ensemble approach, and see if they look, systematically and comprehensively like they'd actually hit a target. And so, so the first challenge was to find all the excipients and classify them. The FDA has this thing called the Inactive Ingredients Database, IID. You can find it online. We found it not well suited to sort of a research purpose. So we created our own version, and that you can, we hope it's easier to search. It's this excipients browser. So if you want to learn more about excipients, you can go to this excipients browser and find the ones, you know, that have been categorized and what they're used for and where you can buy them, and what they're similar to, blah, blah, blah. Anyway, so we found so far just under 700 that are molecular excipients. So like there's honey is an excipient, for instance, according to the FDA, and that's not useful chemically. And there's combinations of things that are hard to deal with, blah, blah, blah. But there's just under that we found so far about 700 simple molecular excipients, and we've got them in this library. We took each one of them and flew them against this, against the library of chembal targets, so ligands characterized in chembal as active for different targets, and put them through this whole process. And the thing was, well, we'll predict which excipient might be active on a particular pharmacologically relevant target, and then we'll test them experimentally. And so I'm going to show you the results of that. So this is propyl gallate I'm showing you here. It's an antioxidant. It's a widely used excipient. It's in a bunch of different drugs. And when you put it through this prediction method, it predicts that the similarity ensemble approach predicts that catechol O-methyltransferase is a likely target for propyl gallate. And you can, so what I'm showing you is propyl gallate as the top molecule, and these are the two known binders to propyl gallate. So these guys are known to bind to propyl gallate, and you can see without thinking too hard why the method thinks that, you know, propyl gallate looks like the ligands of the known ligands of catechol O-methyltransferase. Catechol O-methyltransferase, I should say, is a drug target itself. It's one of the enzymes that ultimately turns off catecholamine signaling by chemically methylating catecholamine, so they're inactive. And I'm showing you along the bottom just some other known catechol O-methyltransferase inhibitors. So once you know that these molecules inhibit catechol O-methyltransferase, it's not a big leap to think propyl gallate will. But the thing about this method is that it can do these comparisons comprehensively. And so this is the E value that I was talking about before. So it's a strong prediction that propyl gallate will be active on catechol O-methyltransferase, but whether it actually is or not and how much it is, that you have to test for it. But can I just say, you know, that this is a weird ass molecule to be putting in your body, right? I mean, this is a medicinal chemist looking at this molecule is going to think, oh my God, that's pretty reactive, and it is. Okay, anyway, so here's the dose response curves for propyl gallate. So it's on catechol O-methyltransferase. It's, you know, like a 10 to 20 nanomolar inhibitor of catechol O-methyltransferase. So that's in the region of drugs. Like the median activity of drugs on targets is about 10 nanomolar. So this guy is quite potent on catechol O-methyltransferase. It's okay. So that was the most potent molecule we found, but we found a lot of others. And this is, I don't expect you to take in this table. It's hard to read. I'm sorry, but it just says that there's a bunch of different excipients. There's sort of 2 columns worth of them here, active against a bunch of different targets, with activities in the, you know, ranging from, you know, okay, 15 nanomolar for propyl gallate to you know, 700 nanomolar for this dye, and you know, micromolar for some of these other molecules. And, you know, they get into the micromolar range and think, okay, well, micromolar, you know, not so bad. But the other thing to say about excipients is that they're often in much higher, they can be there in much higher concentrations than the actual drug is. And so I'm giving you the max, so-called max dose thing. So that's the amount that they occur in the different drug formulations. So in Prevacid, there's 40 milligrams of FDNC red number 40 in Prevacid, and you know, it's a 5 micromolar inhibitor of this transporter. Okay, so this work was done in collaboration with Laszlo Urban's lab. I should have given him credit before. He's a sort of longtime collaborator. He runs the molecular tox group at Novartis in Cambridge in the U.S. And it turns out we had collaborated on some earlier studies, also using the chemoinformatics, and the collaboration had sort of died out. And he invited me to speak at a symposium, and we were having dinner together. And we said, "Oh, so what are you doing recently?" And I said, "Oh, well, we have this funny project on excipients. I mean, I know it's really weird, but," and the blood drained from his face when I told him that. And he said, "Well, we're working on exactly the same thing." So anyways, we decided to collaborate. And what they did is, whereas we went computationally to prioritize things, they went sort of systematically through and tested like the top 100 excipients, according to them at Novartis, against their off-target panel. So this is something that they always do early in compound advancement, is look for off-targets. And so what's shown on so these are all Novartis data here from Laszlo's group. So what's shown here on the y-axis, on the the left column, are the excipients, and what's shown on the rows are their activity on these different targets that are listed at the top. And wherever it's green, it means it's inactive. So a lot of excipients are inactive on a lot of targets. But wherever it's red or not green, it means it's quite active, micromolar or submicromolar. So just to show you a few, so this is FD&C red, erythrocene. It's another excipient dye, and it's 700 or 800 nanomolar against the M1 muscarinic receptor, so it's going to have a dry mouth side effect. And just to show you, these are the dose response curves that they got at Novartis, so that the data I think are pretty solid. This is a sort of more alarming result for several reasons. This is thimerosal. It occurs in vaccines. It's what got the anti-vaxxers agitated, and I really concerned about giving support to the enemy, sort of thing. But it's I never until we started this project, I didn't realize that thimerosal was a mercury-containing compound. It's quite potent, actually, on the dopamine receptor. It's about 150 nanomolar on D3 and similar activities on the D5 dopamine receptors. And it gets into the brain. I mean, that's not our work. It's other people's work show that thimerosal gets into the brain. So these dopaminergic effects might be relevant. At pharmacologically relevant the concentration is relevant to its dosing. It gets into the brain. And these IC50s are a little bit above or a little bit worse than the concentrations it gets to in the brain, but based on primate studies, but not much. So that's actually a bit of a concern. And so that's one reason we were concerned about it. The other reason is that we don't want to alarm people, because we're certainly not saying that people should stop taking their vaccines. It doesn't occur in Europe anymore at all. In the US, it's being banned from most things, but it still occurs in multi-dose formulations that you can get in the US for influenza, for instance, influenza vaccine, and it's still used in the developing world, both for adults and children. This is the last molecule, and I think basically the last thing I'll show you. So this is benzthonium chloride is the bigger molecule I'm showing you here. It's also used in an injectable formulation, just like thimerosal. It's used with ketamine, which is shown on the right here. So ketamine is used for pain and anesthesia as an injectable in hospitals. And it's formulated almost always with this benzthonium chloride, which is an anti-infective agent. And the thing is that benzthonium chloride has a strong hERG signal. It's 500 nanomolar on hERG. And sure enough, this drug, ketamine itself, is not active on hERG. But the excipient is the injectable excipient, and sure enough, this formulation has cardiac adverse effects in the adverse effect literature, like and databases like FAERS. So we suspect that to the extent that this formulation has problems, it's because of the excipient, not because of the primary drug. So that's another concern. So I think, you know, I'm certain I'm out of time by now. So I think I'll stop here, and thank you all for your patience, and take any questions.
Oh, sorry, Shoshana, it looks like you got muted this time. You've been unmuted here.
Thanks, Brian, for this fantastic excursion through your work, and especially this expedient analysis is fascinating, somewhat worrisome. So I will try to look at the questions and oh, we have no questions. Is that normal? Hello?
There appear to be no questions so far. Yeah.
Wow, okay. This is disappointing. Any questions that people want to ask? Can they speak? No.
We do have both the chat and the Q&A panel available to attendees. If anyone wants to type out a question, you can use either one at the moment.
I see just one chat from Diane Kovac. And I don't want, you know, one message. I don't see any questions at all. People are too shy. What's happening? Oh, yes. Now we have question and answers. Thank you. Okay, I have a question from I can't pronounce the name, and Jessica, something like that. Sorry. She asks, does that mean that Novartis will work on changing the formulations?
Yeah, great question. I think, I don't know. I think Laszlo got a certain amount of blowback from his colleagues when, you know, because they had to, you know, he had to get it formally funded within Novartis. And there, I think there was a group of people there who thought, you're just going to create problems for us, man. But he was supported all the way up ultimately. So I think, I don't know if they will. There's another question though, which is, will the regulatory agencies, will the EMA and the FDA start to think about how, you know, what they're doing with excipients and how they change that? And I think, I feel there's two things. One is, you know, a lot of these excipients could be swapped out. Like, we don't really need to use benzthonium chloride, or certainly not thimerosal as an anti-infective. And so I think there's a lot of work that can be done there. But the other thing is, is that it's been a dead field. Excipients have been a dead field for decades. It's really hard to get a new excipient approved. It's almost as expensive as a drug, which is nuts. And the thing is, they're important. Excipients are important because formulating what you discover as you start to take molecules into animals is that they're dosed really high, right? Like just in the injectable stuff or in the formulation you're going to take, the concentration of the molecule is really high. And when it hits the stomach, it's really high. And it's hard for it to stay in solution. So you need the excipients there. The formulation, it's like this dark art. And so the other side of it is it'd be great to get both better excipients, like remove some of these bad actors, but also improve how we make it easier for people to innovate in excipients. And I don't know how that happens, actually.
Brian, do you know whether generic drugs are using similar excipients, or are they changing these excipients?
Yeah. Super important question, which I meant to touch on. So generics, it's a real problem, actually. They can swap among excipients. So like the discoverer company, has a particular formulation that they've tried out, blah, blah, blah. And then when it comes to generics, the generics can swap in a different excipient. As long as they show that that excipient doesn't change the exposure of the drug. Mm-hmm. And the assumption behind that is that the excipients have no activity on their own. Mm-hmm. But they can.
Yeah, they have.
Yeah. So
okay, I have a number of more questions, which which is nice. So Raj Varijapalli says that it's a fascinating talk, and he asks, I wonder if these results can be fed back into mechanistic models.
So I think Raj is probably asking about the SARS stuff, and yeah, we hope so. Yeah, I think that's actually that's the soft underbelly of repurposing is mechanism, and I think with the proteomics we at least we know the targets, and our challenge now is to go deeper into mechanism, and we passionately want to do that and are trying to do that, and exactly how you do it depends on what in the individual targets. But one big way is the chemical genetics, knock down the target, see the effect on if the molecule still has an effect. And that at least establishes the target, and then you can follow it downstream. So for instance, looking at the phosphoproteomics as the molecules affect signaling. So those are things we're doing.
But Brian, in the PPI study, in the PPI network, you only have isolated viral proteins that interact with the human host. What can you comment on? How do you think this may change the picture that you are analyzing?
Oh, for sure it changes, because we know that trivially, we know that some of the viruses operate with other some of the viral proteins operate with other viral proteins, which of course are not there in that study. So it for sure has an effect, but it's tricky to do it properly because, working with SARS-2 is a beast, biosafety lab 3 containment. So it's very restrictive. So the proper way to do it would be with, full viral infections or somehow attenuated viral infections. It's a little bit tricky.
But you could add other components that already from COV, from COV analysis that they do interact together anyway. I think that's an important point.
Yeah,
we have more questions.
Yeah.
Sorry, go ahead.
No, I was going to say that's the next step. I agree.
Mm-hmm. So there's another question. I think Raj wants to clarify his question. So I'm just reading it, and if it doesn't bring anything new, we can skip. So he says, what I mean by this is, in mechanistic model, is the primary drug is used and tested. Should we be considering the whole milieu of the formulation with the excipient activities in the mechanistic models, which is fair question.
I think it's more important. Well, if you mean by mechanism, I think it's important for sure in animal studies to understand this. In terms of the primary actions of the drugs, I don't, on the disease, I don't think it'll be rare that the excipients affect that in an important way. But in understanding the off targets of the drugs, what we say, oh, this is, such and so drug has such and so off target. It's mostly based, unless you do the molecular studies, it's mostly based on these whole human phenotypes, and there the excipients can be an important confound. And okay, let me tell you one other thing about this that makes it more complicated and scary is, most drugs are taken by people over 65, right? The primary consumer for the pharmaceutical industry are the elderly, and they usually take more than one drug. And so they're not just taking one set of excipients, they're taking combinations of excipients.
Yes. That's terrible. So Anichka is also asking, you know, does that mean that excipients can be potentially considered as new drugs? Which is, she says, is this good or which is good? What is your answer to that? We lost you?
No, I'm here.
You're here.
I think they can be a new, I wouldn't think they're drugs. Most of them don't have the right properties for drugs, just looking at them, although that didn't come through in my seminar. I think they can be good, like initial molecules to like when people are thinking about repurposing and I would say don't forget about excipients because they are approved molecules. They're approved for human use. So in that sense, yeah, I think she's onto something. Yeah.
Okay, so there is another question about the excipients, you know, that has really motivated everybody, worried maybe everybody. Has there been, that's a question by Ronald Rodriguez, Santiago. He says, has there been any work on combination of excipients and their activity?
Not much, unfortunately. And they're always used in combination. It's rare to find only a single excipient in a drug. And you know, they're approved. It's funny, you know, the FDA hasn't, says, okay, you can only use an excipient this much in a day. So that's what their dosing information means. It means you can only, if you're going to take the pill 3 times a day, well, you have to be aware of how much of excipients are in those 3 pills, for instance.
So the dosage is based on excipients as well.
Yeah, but they don't, but then, like I say, you know, like my mom, for instance, takes 7 drugs every day and they don't, and the same excipients can be in them.
Oh.
And they don't control for that.
No, okay. Yeah, some doctors don't do it either.
Almost nobody.
So another question is now about SARS-CoV-2, you know, how to consider, that's by Raj again, how to consider SARS-CoV-2 mechanism when there are so many targets in the interactome. Yeah. You have to deconvolute.
Yeah, it's complicated. I mean, it's as almost as complicated as a whole cell. You know, they have to replicate and they have to you know, put themselves back together again. They have to get out and they have to suppress the immune response. And so there, for, you know, these 29 gene virus, you know, does a lot of amazing things. That's very complicated. But you can break it, but you know, it's like anything else. You can break it down, right? So the NSP6 and some of the other viral proteins are involved in these formation of these dimembrane vesicles, and they recruit human proteins like sigma-1 to help with that. And exactly what every one of those guys is doing is, I'm not sure, but I think that's the one with the proteomics helps in that it makes a very complicated, you know, 332 proteins. You think, wow, but you can break it down into components.
Do you know of any of the viral proteins that are associated with proteins that cause the cytokine storm?
I don't.
Or
I mean, I because I'm not an immunologist, my trivial explanation is that's just the, you know, the immune system.
The reaction immune system,
going wild. And you know, that's of course, you know, what, you know, dexamethasone is doing is it's suppressing.
Right. Yeah. And steroids also, so all kinds of steroids.
Yeah, a lot of other steroids too.
Yeah. So Raj is now talking about, asking about, would whole cell models be useful in this regard? Well, you more or less answered, you know, this issue already, but go ahead.
I mean, for sure.
So I mean, one question by, sorry.
Go ahead.
Okay, one other question is by Lindley Darden. To all the participants, all the panelists and participants, are you willing to predict when an effective drug for SARS-2 will be found and also made available for use?
Oh. Well, I can predict the past. I mean, you know, remdesivir, I think, has shown some efficacy, and dexamethasone has shown some efficacy. I mean, in clinical trials, and I think certainly remdesivir is being widely used, and I bet you dexamethasone will be too. But in terms of, you know, do we have a molecule that will really change things? I don't think remdesivir or dexamethasone has on their own really shifted us to a new place. When that will happen, it's hard to say, but I mean, I think one of the great things, there's so many terrible things about what's happening now, but one of the great things is so many people have thrown themselves at the problem. It's really encouraging, and there's, especially the drug companies, I gotta say, those are, you know, ultimately the molecules will probably involve drug companies, and I think they've really thrown themselves at it. If there's a problem, it seems chaotic to me. I mean, there's because of a failure of politics.
Yeah. That's
certainly in the United States, that it's a bit disorganized.
But do you think a drug will come before the vaccine?
Well, yes, of course, because they already have. And it's a question, when you ask, will a drug come, I think you mean, will a drug that really changes things?
Yes, exactly. That's my question.
I don't know. I mean,
how to say? Yeah. So much is thrown at the vaccine right now that, yeah.
Yeah, but
let's hope for one or the other, and maybe both.
Yeah, but here's what I would think, sort of to think about is that, this isn't the last time. We've had 3 coronavirus outbreaks in the last, 17 years.
Yeah, 17 years. Yeah.
So there's going to be another one.
For sure.
And how do we prepare ourselves for the next one? We should have an infrastructure in place, so we know what we're doing, and we know what all these steps are, and it's centrally organized, and we're able to marshal resources, and we've done some of the preliminary research, and can enable, we're still involved, for instance, in new molecule discovery, not just repurposing, and it's too slow, I got to tell you, but there could be ways to speed it up, and I think we should start thinking about that now, while our minds are focused on how important it is.
Yeah, we should continue to be motivated and go ahead.
Yeah.
Yes, I don't know if there's any other question. Oh, yes. Yeah, two other questions, and then I think we'll stop if you're not too tired. Raj is saying, going forward, is the present experience and the learning going to change how fast the drug discovery process can be executed to other diseases? Are the major bottlenecks still remaining? So, yeah, many bottlenecks remain, and some hopefully will be lifted.
Yeah, that's my answer, basically. Unfortunately, all the same bottlenecks remain.
Yeah.
And they're there for a reason, actually, many of them. But I think we can remove some of them, and it's a social issue as much as a science issue, and I don't have any answers for Raj about exactly how to do it. I just feel like it's super important that it be done, because there's going to be COVID-24 or COVID-27 or something, and we're going to be right back where we are today, and that'll be catastrophic, and we need to think these things through, and I don't have any answers. I just know that we need answers.
Yeah, collectively we need to go forward with this. So another question is about the Russian vaccine, and there is many different aspects about the Russian vaccine and Russian vaccinations which have come up recently. So the question by Farzan Noziri is, have you heard about Russian vaccine that they are testing right now? They have taken adenovirus as the container to SARS-CoV-2 coronagene to transport virus to cells. What is your take on that?
I don't have an opinion. I'm sorry. I'm not. Yeah,
Yeah,
I'm just not expert in that area. Anything I would say would lower the sum total of human knowledge.
Yeah. And then the most recent finding is that if you vaccinate against polio, you can get vaccinated also against all kinds of coronaviruses and influenza as well. That has come up, you know, very recently. So it's also an immunological, you know, story.
Right.
Shall we go to the last question? I think we should stop after that. That's again from Raj. This presentation discussion has been very educational, illuminating, and motivating. Many thanks. So this is what I was going to say. Thank you so much for your effort, your talk, and I hope you continue this work and help us all get out of this mess.
Thank you all for attending.